All right, we'll pick up where we left off. I apologize. The software that we're using that's provided can only handle 15 minutes at a time, and maybe some of you can only handle 15 minutes at a time, so it's not necessarily a bad thing. But anyway, we were still working on this slide and talking about the layers of the tube system. Now, there all of the layers aren't precisely the same all the way through, because you have different things happening each different way. But the actual canal begins in the mouth and the throat, and you have these layers in the mouth and the throat too. They're not quite this spelled out, but areas where you have more friction or you have more movement, they're going to be designed somewhat different. And certainly in the mouth and the esophagus, you don't have that connection to the visceral or to the parietal peritoneum, so you have other things that are actually covering the outside of those layers. But we think we did the submucosa. So we've introduced some blood vessels. Again, those are going to be the blood vessels. We introduced the blood vessels in the mucosa, more in the submucosa, and lymphatic vessels are going to be the areas where things are absorbed. Also, glandular tissue. This glandular tissue, there's always going to be a need for lubrication, and the glandular tissue helps to control and regulate what the speed, how this bolus is moving along, communicating to whatever it's moving to, so the area is moving forward to. So you can think of these glands as secreting chemicals for communication within the GI tract itself. Actually, you have some people who say that the GI tract has more innervation to it than any other area of the body. They kind of call it the third nervous system is the enteric nervous system. I think we talked a little bit about the muscularis externa. I know I remember that because I apologize for that capital E. And what happens is you have muscle that's going to move this along, but there's also muscle that actually needs to reform the shape and get it ready for the next piece of shape to come along. And then you have the serosa, which is the outer layer. I think the place that we really left off was the nerve plexuses, or plexi, whatever the plural of that Latin term is. I guess it's not a Latin term, but however, a plexus, I guess. They're innervated by the autonomic nervous system. Remember, that's the nervous system that you don't control, consciously control. It has two divisions, sympathetic and parasympathetic, and you probably remember the sympathetic is your flight or fight, parasympathetic is your dream and digest. Well, both of them have innervation in this area, but it's actually the parasympathetics that are what we generally want to control our GI tract. I mean, we want that parasympathetics to control it because it's the one that's going to move things along. There are two areas. So you have the mesenteric plexus that innervates the smooth muscle in the muscularis externa. So those are, remember, if you go back to number 3, that's the two layers that are going to actually create that peristalsis. That's innervated by that myenteric. And then you have the submucosa nerve plexus, which your book calls, I'm looking that up, Meissner's plexus. So your book calls it Meissner's plexus, and that controls glandular secretion. So that would be a local control. And then you can think of the myenteric plexus as a extrinsic, so something that's coming from the CNS. Here you finally get a picture. You have a nice picture in your book, and you have the layers from deep to superficial. Deep, you have that mucosa. When you think of the word mucosa, you're thinking of mucus secreted. It's actually epithelium with connective tissue. You have blood supply in there because it's living tissue. You're going to have blood supply all the way through the whole thing. And you do have a little bit of smooth muscle in that one. And then you have the submucosa there. Submucosa, remember that's where you have your first nerve intervention that mesenteric, that Myers. Let's see what they call it on here. I don't see him calling it anything on here, but the submucosa, that's where you have the Meissner's plexus, so that's the glandular release. And then you move out to the muscularis externa. That's a pretty thick one where you're having at least two layers of muscles, longitudinal and circular muscles that are going to create that movement called peristalsis. And then you have on the outside the connection to the serosa, which is connected to the parietal peritoneum. It's kind of the capping of all of this. You're still going to have connective tissue, but it's kind of the capping over all of this. It's a nice picture. And this is the wall of the GI tract. Generally, the entire wall has this same basic design with some modifications based on where you are. Another picture of it, just again, I'm repeating things because one picture may work better for you than another. This one happens to be the one that's closest to what looks like in your book. You also have, in this one, you have your first picture of the pila, the plica, yeah, the P-I-L-C-A, plica. I guess that's how we're going to pronounce it. And that's how it's pronounced. Don't get me wrong. If you come up with a better pronunciation, you let me know. And what happens with that is the small intestines are about 20 feet long is all. But because you have these little peaks that come up, and you can see them if you look, we're on the right side, if you look at the top right picture, excuse me, you have these actual folds, and on top of all these folds you have little these little plica pushing out to increase surface area. The big thing to remember about this, and please don't remember how difficult it was for me to pronounce it, but remember that what these do is increase surface area, because that's the important part. The more of them you have sticking up, the more surface area that you've created there in that small intestines. The more surface area, the more likely you're going to be able to have absorption take place. And they're so designed because what you can then have is you can have your blood vessels in them, and then you can have your lymphatic vessels, as well as you can see your layers of the that we just talked about, the muscularis externa, the serosa, the submucosa, and the mucosa, all the layers we just talked about, as well as the nerve innervation. Here's a nice picture of peristalsis. It's really kind of cool if you get the picture. You have a bolus, which I guess we're in the small intestine, so we're talking about the chyme, but it's kind of, well, peristalsis happens in the esophagus also. What happens is you have a contraction of the longitudinal and circular muscles. And particularly the circular muscles are contracting and making sure there's a door so this particular bolus can't move backward. And the longitudinal muscles are actually holding it tight. And then what you have is on the high side of the that contraction that's holding it in place, you have this kind of wavy motion go on. So basically what it does is it kind of shoots it forward like it's shooting something out of a cannon. So it shoots it forward. And it does that moving it along all the way through the whole GI tract. And that's the portion that has to get back in shape, getting ready to hold the next one. So that's peristalsis. It's one direction, and it moves the food along. Segmentation is also taking place. This is moving the food back and forth. Moving it back and forth so that it can mix, moving it back and forth so it can come in contact with the surface area, and then as that particular what really is not food any long, those macro molecules, which are well on their way becoming micromolecules, can actually be mixed with the fluids and give it a chance for the nutrients to be absorbed into the bloodstream. Peristalsis, segmentation, two different types of movements, two different reasons for types of movements. And then here you have, we've talked about this a little bit, about the beginning of the regulation, very start of regulation of the GI tract. This is just the tube. Just acclimate yourself. You have your serosa, you have your muscularis externa, you have your submucosa, you have mucosal layer, and what you have are the myenteric, remember that's in your muscularis externa, and then you have your Meissner's, which is in the submucosa, and the Meissner's, which is in the submucosa, this is going to release, facilitate the release of these hormone or these chemicals that are going to regulate the environment. So the pH, regulate the actual chemicals that are happening, so release acids, buffers, enzymes, so the chemicals that are needed at that particular moment to change pH, to stimulate the muscles, or to stimulate chemicals to be released. And then the myenteric, which are in the externa, are under the control of the CNS. So the long reflex is under the control of CNS. Short reflex is under the control of what's happening in that specific spot. That's what we're talking about, stretch receptors and chemoreceptors. So you have receptors that are intrinsic or in that spot controlling the muscles. You also have the CNS controlling muscles, so you have an extrinsic. And so this picture's in your book too. So a little bit more about the actual regulation, neural, hormonal, and then local regulation. Moving into a little bit more anatomy, you have your oral cavity and your pharynx. I won't mention all of these parts of the oral cavity, but you do have you do have the teeth with regard to mastication, tongue for mixing, tongue also for identifying whether something has glass in it, so identifying texture, hopefully not glass, but identifying texture. A lot of times I eat nuts, and sometimes you have the shell of the nut in there, or if you've ever eaten eggs, you have the shell of the egg. Your tongue picks that up right away. So not only for taste buds, for taste, but also mixing and also the feel of the food. And then you have a sagittal section where you have the tongue, and it actually is attached to the mandible. It's attached in the front. Doesn't seem that way. Has a back attachment too, I should actually say that. And then you have the area where you have the oropharynx and the pharynx in terms of the oral cavities, the beginning of it. You don't have to know all of this, but I'd be kind of familiar with these pictures, certainly for lab. And then you have your salivary glands. I think the thing that you want to know about them is their existence. So the parotids, submandibular, and sublingual. And the parotid salivary gland up by the ear. Submandibular tends to be the lowest on the chin, so below the mandible, and then sublingual is below the tongue. I know we have a nice model. Sometimes it's difficult to tell the difference between the sublingual and submandibular if they're not both there. And then teeth is where we're going to pick the next one up. These things go by awfully fast.